Hello everyone, and thank you for coming to the penultimate lecture of our International Studies Institute lecture series. We have one more on Monday at 12 o'clock in this place by Delia Katara, discussing gender issues, education, and domestic violence and their relationship. But to step back, first of all, I'm Hélène Bastel, the Director of the International Studies Institute, and Associate Director Christine Zauer is right there. She directed ISI for about 5 years before I started, and is also the main advisor for our undergraduate major in international studies, and we have over 200 students majoring in that. The International Studies Institute started about 10 years ago, and Marisa Bokovoy, professor and chair of the history department, was the founding director. And this tradition of the lecture series started from the very beginning. A series of lectures on a specific theme every fall, targeted primarily to upper level undergraduates, but of course also to everyone else, the general public, and so on. So this year's theme, Modern Societies in Crisis, Global Challenges and Solutions, started as an idea about a year ago. During the last 5 years, we have witnessed unprecedented upheavals that have pitted the principles of democracy, multiculturalism, and respect for human dignity against often intransigent political and religious positions. Concurrently, the economic crisis affecting several countries within the European Union has caused increased unrest within and beyond the EU. Speakers have been addressing the crisis of modernity from the local to the multinational levels, comparing developments around the world, and examining problems and proposed solutions, and some of these solutions are coming from the quote-unquote less developed parts of the world. Some of the problems are coming from the first world parts of the world, and the idea is to actually look for possibilities for a better future. Modern Societies in Crisis, Global Challenges and Solutions, we hope has been enhancing our community's understanding of current political and economic upheavals and their effects on New Mexico and the Southwest. The lecture series has included actually 5 UNM faculty, and Stephen Bradfute, today's lecturer, is one of them, one of us. Also 3 New Mexico experts and activists, as well as 3 US-based scholars, and the preceding lecture by Nadia Kravitz was an example of one of those. Nadia came from Harvard, where she's a postdoc. We also had 2 international speakers, one coming from Canada, one from China. And I really want to thank the speakers for making this possible, for participating, for sharing their research, their energy, their questions, their time, and ideas with us. I have to say, when we started, we didn't know that Ebola would be one of the crises, and actually it was Christine who said, we have to have a lecture on the Ebola crisis, Eleni. And after a bit of searching, we were honored to have today's presenter with us. I also want to thank the various groups that supported this lecture series with financial resources, with ideas, and moral support as well. Within the university, that includes the Office of the Provost, the College of Arts and Sciences, the University College, the Feminist Research Institute, the History Department, Peace Studies, and I'm not forgetting anyone else. And I think that was it. And then beyond the university, the Colorado European Union Center of Excellence, the European Union, as well as the New Mexico Humanities Council and the National Endowment for the Humanities helped make this possible. And all along, this lecture series has been videotaped by a very, very competent and professional crew that come under the general name of No Reservations Company. And it has been also a pleasure to be working with that. The International Studies Institute is really a team of that works together very well. I mentioned Christine, also it includes Julie Williams, who is our graduate assistant, Ken Carpenter, adjunct instructor in international studies, and Jasmine Knight, who is our operations specialist, makes everything work well. Now, for today's lecture, Ebola virus, past, present, and future, I would like to introduce Stephen Bradfute, Research Assistant Professor of Internal Medicine, Center for Global Health at UNM. Stephen specializes in hemorrhagic fever viruses. His current project is trying to develop a pan-Ebola virus vaccine. And thank you very much.
Thank you.
Ears.
Appreciate it. First of all, thank you for the opportunity to talk about this virus. A little background about myself. I grew up 4 hours east of here in Portales, New Mexico. Got my bachelor's from Eastern New Mexico University, a Ph.D. at Baylor College of Medicine in Houston, Texas, in Peggy Goodell's lab. And then I really wanted to work on infection and immunity. So for 6.5 years, I was a postdoctoral fellow at the U.S. Army Medical Research Institute of Infectious Diseases, or USAMRIID, in Fort Detrick, Maryland, where I worked on Ebola virus for about 6 of those years. And so then I moved back to UNM a couple of years ago, and I've picked up my work on Ebola from there. So what I'm going to try to talk to you about is Ebola virus, how it was discovered, what we've known about it, also talk about the current outbreak and why this is different from other outbreaks, and then what the future holds for Ebola virus containment and research. And don't let the tie fool you. I'm a pretty informal guy, so if you feel free to ask questions during the presentation. I'll also try to save some time at the end for questions. Okay, so I kind of structured this talk based on a lot of the questions I've received over the past several months as far as what people are interested in. So I'm going to try to answer those general questions, and then hopefully from there it'll generate further discussion. So what is Ebola? Well, the history of Ebola Ebola wasn't discovered until 1976, but this type of virus, the history begins in 1967 in former West Germany, in Marburg and Frankfurt, and then also in Belgrade, what was then Yugoslavia. Lab workers that were working on development for polio vaccines started becoming sick. And these workers, they imported monkeys from Uganda, and they would process their liver to obtain cells to generate polio vaccine. They started becoming sick, and originally they thought it was a different virus like yellow fever. But they had an interesting disease progression. They started out with fever and headache and general malaise. They just didn't feel well at all. Then that turned pretty rapidly to vomiting, diarrhea, and nausea. And they developed this interesting rash that was over large swaths of their body. There was some external bleeding. It wasn't in all the patients. And they weren't responsive to antibiotics. So antibiotics are effective against certain types of bacteria, but not against viruses. A total of 31 individuals were infected. 7 of those 31 individuals died. 25 of the 31 individuals had direct contact with monkey tissue. So it didn't spread very much beyond those that were directly involved with the monkeys. And so it is amazing. You can read these papers. They're originally published in German in 1968. If you read the original papers, with remarkable speed, they identified a new virus, and they called it Marburg virus. And as you can see here, this is a picture of the virus, and it's kind of stringy. It looks like thread, so they named it a filovirus, philo being Latin for thread. So these are the filovirus family. Marburg was the first one discovered, as we just discussed. And in 1976, there was a similar outbreak in the former, what is now the Democratic Republic of the Congo, then it was called Zaire, and also an outbreak in Sudan. And the symptoms were very similar to Marburg. The virus looked a lot like Marburg, but it was a completely different virus. and they named it Ebola virus because of the proximity of the outbreak to the Ebola River. So there's actually 5 different types of Ebola, and these types, we call them species. Until recently, this has been a fairly scattered and not very prevalent infection. Of course, the current outbreak has changed that. But over time, there's only been about, between Ebola and Marburg, only roughly 12,000 infections that we know of. The lethality rate really varies by the virus species we're talking about. So the virus that's currently causing the outbreak is called the Zaire virus, and it was originally discovered in '76. Marburg virus actually is equally lethal, if not more lethal. It just doesn't quite get the publicity of Ebola. How does the virus spread? Well, it's thought that in Ebola virus, it replicates in a certain fraction of bats, a certain type of fruit bats. And it can replicate in these bats without causing disease, and then it can spread from bats to monkeys, or bats to humans, or bats to monkeys, and then from monkeys to humans, and then from human to human. That's the theory. It's known that Marburg virus is carried in fruit bats, and it's suspected for Ebola. Accidental hosts. What I talk about with this is generally it's not in a virus's best interest to destroy its host. So the virus naturally replicates probably in bats, doesn't cause much harm. And then when it jumps into other species that are very susceptible, these species are, of course, humans, non-human primates, and monkeys, gorillas, chimpanzees. The duiker, which is a forest antelope, porcupines, was actually been found to be killed with the virus. And there is one type of virus that's not pathogenic in humans. It's called restin Ebola virus, and that's found in pigs. Endemic areas, generally, it was Central Africa around rainforests, and then the non-pathogenic Ebola virus in the Philippines and China. And of course, since March, it's had an outbreak in West Africa, whereas Marburg viruses tend to be found in Central Africa and in arid woodlands. The transmission, which we'll get to in detail in a bit, is close contact. I'd like to bring this up. A few years ago, there was a newly discovered filovirus called Cueva virus, and they discovered it because there's all these bats dying in Spain. And they've found that there's a related virus to Ebola in these bats in Spain. What they don't know is whether or not it's pathogenic in humans, because no one's isolated the live virus to be able to do lab studies in non-human primates or animal models. So as I mentioned, there's 5 different Ebola virus species. Zaire, which was first discovered in '76, is originally in the Democratic Republic of the Congo, or Zaire, back in those days, Congo, Gabon, and of course the current outbreak in red here began in West Africa and has spread Guinea, Sierra Leone, Liberia. It's also spread to Nigeria, Senegal, Spain, the US, and as of last night, Mali received its first case of Ebola from this outbreak. There's been a total of 17 outbreaks in humans of Zaire. Sudan Ebola virus was also discovered in 1976. The lethality rate's about 54%. I want to say here, the Zaire species, it depends on who you talk to. The numbers that we're getting right now are not, we're not sure how accurate they are. Some people say the lethality rate of this Zaire outbreak is about 50%. Some say it's closer to 75. Historically, Zaire lethality rate has been closer to 80%. The newest species of Ebola is Bundabujo, and there's only been 2 outbreaks, and it has a lower lethality rate of about 32%. There's only been 1 instance of Thai forest virus. It's known to destroy, unfortunately, chimpanzee populations, and there's been 1 infection of someone that was working on dissecting a dead chimpanzee to figure out why it died, and they contracted the virus and survived. And then restin Ebola virus, which you might know from the book, The Hot Zone, has been imported multiple places in the world, but it's not known to be pathogenic in humans. So it really kills monkey populations, but there's been 11 documented cases of the virus getting into humans, and none of them got sick. So, as I mentioned, the Ebola infections have been pretty sporadic over time. This is a documentation of Ebola virus outbreaks beginning in 1976. And you can see that there's a couple of outbreaks here, and then there's a long period of time where there's fewer outbreaks, and it kind of comes and goes. And in general, not a lot of individuals have gotten sick. And you can tell this is a few months old. The current outbreak has reached about 10,000 individuals now. So in the past, Ebola virus has been occurring in very isolated areas where it's easy to contain, or not easy to contain, but easier to contain. And then eventually it burns itself out, the infection goes away. And what's going on now is we're getting, for the first time, virus outbreaks in West Africa in very, very large cities that have very poor healthcare infrastructure, and so it's spreading very significantly. So one of the questions I get a lot is, why is Ebola more dangerous than other viruses? Well, this is this is an interesting question. To our best knowledge, I mean, I'm a scientist, so I don't like yes, no. I like to say qualifiers. To our best knowledge, Ebola is really good at blocking an early part of your immune response called type 1 interferon. And basically what this arm of the immune response does, it shuts down viral replication. It limits virus replication until the rest of your immune system can come and combat the virus. Ebola virus has 2 different proteins that are really good at blocking the ability of your virus to initiate the type 1 interferon response. And so when that happens, the virus is able to replicate and evade the early immune response, so it gives it a leg up. The virus is also very fast replicator. So within a matter of days, you can get infected with a few or maybe 1,000 virus particles, and then within a few days, you might have 100 million for every milliliter of blood. So it replicates extremely quickly, and it can cause organ damage in the cells in which it replicates, and then it can induce overinflammation, which can be harmful too. So together, that makes it a very, very dangerous virus. So here's a model for pathogenesis of Ebola virus infection, or kind of a graphical representation of why Ebola virus is so dangerous. As you can see here, this is a terribly drawn Ebola virus. I am not an artist. So the virus, we think, usually circulates in bats, and then it can hop into monkeys or humans. And so say the human gets it you can get it from a number of ways. You can eat contaminated meat, for example, bush meat, monkeys or forest antelope, and it can be contaminated with the virus. You can get it through cuts and then coming in close contact with the virus. And needle sticks from sharing needles, that sort of thing. Close contact is the most common way it's transmitted. And so the virus initially enters cells of your immune system, certain immune system cells, and it starts to replicate in them. And usually at this point, the immune cells make your early type 1 interferon response that blocks viral replication, but the virus blocks the ability of the host to do that. As a result, it starts to replicate. And when it starts to replicate, your body over-responds by having a ton of inflammation. So you get fever, you can get a lot of blood clotting, and you clot so much that you block off blood flow to your organs, and you can have organ shutdown. At the same time, because your blood clots so much, you can't clot anymore, so you start bleeding. So that's why you see pictures of people bleeding from the mouth or from the eyes. But more dangerously is internally, you start getting leakage you start getting internal bleeding, and you can start losing blood pressure, and you can have shock-like symptoms that are very dangerous. And because the virus replicates so much, it spreads throughout your body. It can infect liver, or pretty much any organ in the body. It infects a wide range of cell types, and it can cause damage to those cells, and then it can really decrease liver function, kidney function, et cetera. So together, it can give you really, really unfortunate, nasty symptoms. So what are the clinical signs and symptoms of Ebola? I always laugh because sometimes you see a headline, "Man hospitalized with Ebola-like symptoms." Well, early Ebola-like symptoms are just like the flu. So fever, headache, fatigue, muscle aches. Sounds like the flu, sounds like malaria, sounds like typhoid fever. It's followed by diarrhea, vomiting, and rash. And later on in infection, you can start seeing blood with diarrhea and vomiting. And hemorrhage or bleeding, which we discussed, can occur internally. It can occur in your eyes or out your mucosal surfaces. And I mentioned before the blood clotting problems where you clot so you can shut down organ function, but at the same time you use up all your clotting factors so you can bleed. And that leads to multiple organ failure, liver damage, other organ damage. The incubation period is 2 to 21 days. And what I mean by that is how many days from the time you're infected till you start showing symptoms. So in general, 5 to 8 days after you're infected, you start showing symptoms. But it can be as long as 21 days. Some people say maybe even longer. So that's why in the news a lot of times you'll hear people say, okay, it's been 21 days since the last infection, that's a good sign. This is why. If you have the virus, you'll start showing symptoms generally within 21 days. Now, one of the problems is because Ebola virus, we got to, even though it's really, really big in the news and on a lot of people's minds, it's very rare compared to a lot of other diseases that have very similar symptoms. So it's not unusual for Ebola to be misdiagnosed for something like malaria or cholera or typhoid fever, because these diseases are much more prevalent in the areas where the Ebola virus lives. And so oftentimes you'll get misdiagnosis. That's one of the problems with this current outbreak, is it occurred in 3 countries in West Africa that have never seen Ebola before. So no one was really looking for Ebola virus infection. It looks a lot like malaria, which is a lot more common. So I have some pictures. I took out some of the more graphic ones simply because I'm used to giving lectures to medical personnel, and so that's a little more necessary, but I don't want to be show unfortunate pictures just for the sake of showing them. But this is, you hear a lot of people talk about red eyes. Well, you can get hemorrhaging in your eye here with Ebola virus infection. This is disseminated intravascular coagulation, probably, where you basically get a lot of the coagulation I was talking about, and you're clotting and bleeding at the same time. It can manifest like this. These are both from Ebola patients. So, big question, how does Ebola virus spread? And we'll get to the possibility of aerosol spread here in a minute. What we know is that it's through close contact primarily. By close contact, I mean people being very close with each other. It can spread through blood, sweat, tears, saliva, urine, feces, and vomit, because a lot of the late-stage symptoms are diarrhea and vomiting, and it can spread that way. Interestingly enough, after you've recovered from Ebola virus and you don't have it in your blood anymore, and you get sent home, it can still persist, for example, in semen for weeks after you're cleared. So a lot of times people will say, okay, you're cleared, but watch your sexual activity because you can transmit it through semen. It tends to live longer in those. But it's not like HIV where it lives in the body forever. You're going to clear it pretty quickly. So unfortunately, here's some more pictures. So this is an Ebola ward in West Africa, and you can see here, there's a patient here, and there's a lot of probably diarrhea mixed with vomiting, a lot of blood. So when you talk about a healthcare setting, when you have a resource-poor area, you can see the possibility for spread from patient to patient within a hospital. Bushmeat. So bushmeat is a major source of food in a lot of places where the virus is endemic. And since monkeys and forest antelope can be contaminated with the virus, there have been instances of the virus spreading from someone that found a dead monkey in the forest. They butchered it and to eat it, and the virus spread. So it can be caused through ingestion. This is a ward in West Africa, an Ebola ward, and you can see you have patients in very close contact with each other. So maybe someone's in with malaria and put in the same room with an Ebola virus patient, it can spread that way. And also needle sticks. The reason I show this is for the 1976 Ebola Zaire outbreak. It was originally misdiagnosed as malaria, so they were giving shots of anti-malarials to people. Back in those days, you just gave someone a shot with your needle, and you put it in soap and water, and then you gave someone else a shot with a needle. And actually it spread the Ebola virus through injections through all these individuals from a hospital. So why is this outbreak worse than other Ebola virus outbreaks? Other Ebola virus outbreaks have occurred, there's been 20 some odd Ebola outbreaks. Almost all of them have occurred in very isolated areas, in villages, and mostly in Central Africa where there's not a lot of population. The current outbreak is the first one that's occurred in large cities from person to person. So generally the way you can contain infection, but when you have a large city and huge populations, it's harder to do. The affected countries don't have the healthcare facilities or infrastructure to deal with containment. The best way to contain the virus, which we'll get to in a minute, is to isolate patients that are sick, track down the people that they've contacted, and if they show a fever or signs of infection, isolate them. And by isolation, you can really control infection, but that takes the amount of healthcare facilities to hold all the people that have infection and the ability to trace people that they've been in contact with. And Sierra Leone, Guinea, and Liberia don't really have that infrastructure. Even if you don't have a lot of infrastructure, you can contain the virus in smaller areas that are more remote because it decreases the possibility of spread. And as I mentioned earlier, this is the first time in Liberia, Sierra Leone, and Guinea that Ebola has been found. So it took a while for people to realize it was Ebola because it never happened there before. So how do you control spread? Well, we mentioned very briefly, it's boring, it's low tech, and it's extremely effective. We've had 20 some odd infections with Ebola in Marburg, and every single time they've been able to contain it, and sometimes it takes a while, simply by isolating patients that are sick, finding out who they've been in contact with, and if those people get sick, isolating them as well until the virus runs its course. But that's easier said than done. So here's, again, you hear people talking about, oh, patients are getting treatment in healthcare facilities. Well, in West Africa and a lot of places during the current outbreak, this is what it looks like. This is an Ebola ward. You can tell. There's blankets. There's people are rotating in and out here, so it's easy to pass infection on. Unfortunately, this is an individual that died from Ebola in his home, and no one came to pick up the body for a couple of days. So in protest, they dragged him out in the street. And so there's a couple hundred people looking on. The healthcare workers, you can see this individual, he's covered in head to toe in what we call a Tyvek suit that blocks It repels liquids, and he's got, you can see he's got goggles on. But you have to be able to wash this equipment. So here are gloves and boots that are protective. They take them out, they stake them on wooden sticks, they spray them down with bleach, they let them dry in the sun, they get back, put them on, and go around the next day. The virus, even though it's not super contagious, it only takes one virus, functional virus, to cause infection. A lot of other pathogens, you're going to need hundreds or thousands. With Ebola, you probably only need 1 to 10. So even though it's easier to say you can control the spread by isolation containment, like for example in Liberia, I think they're saying roughly 75% of the Ebola infections get sent home because they don't have room for them in the hospitals. So it's a simple infrastructure problem. So this is the type of fruit bat, one of the types of fruit bats that's thought to cause Ebola virus infection. And just to show you the possibility for spread, this purple line is the natural area in which the bat is found. So as you can obviously see, these are most of the Ebola infections in the past, Central Africa, but the bat can range to West Africa where the current outbreak is, and there's possibility of it spreading even further. So it is going to be a worldwide problem. Of course, international travel patterns have, as we're seeing now, have really increased. Over 1 billion people take airplane trips each year. Actually, there's been and even low-tech travel can spread disease. The only case in Senegal was a man that was sick in Guinea, and he just simply walked to Senegal and had infection there. So walking, trains, cars, airplanes, this is, we're a global area now, and viruses can spread rather quickly. So this is one of my pet peeves. There's a lot of talk about Ebola spreading through the air because there's a gentleman who published an article in an op-ed in the New York Times saying that what Ebola virus researchers were worried about but would not say is that Ebola will mutate to start spreading through the air. That is a possibility. We've never known that ever to happen with a virus that's not spread by air to mutate to be spread by air. So I'm not a virologist, I'm an immunologist, so I study the immune response. I asked 2 of my most respected colleagues who are Ebola virologists, and I asked them what they thought of this. The first said, yes, and my Mini Cooper will mutate to become a Ferrari. The second one was more succinct, and his response was, idiot. I'm not calling this gentleman an idiot, but there is very little evidence that this virus is doing that in the current outbreak. Now, if you get up and you sneeze in somebody's face, there's a very good chance you're going to transmit Ebola. But it's not like influenza or SARS, where it's in the air, can go dozens of feet and infect other people. Could it happen? I'm sure that's a possibility. That's really low on our list of concerns right now. The list of concerns is finding the infrastructure to isolate patients. So where did this idea come from? Well, there is some evidence suggesting it could happen. Of course, I mentioned it's not naturally spread by aerosol, but there was a group in, I believe it was 2010 or 2012, that infected pigs with the Ebola virus Zaire. Pigs don't usually carry that particular type of Ebola. They infected the pigs with Zaire. They put them in the same room as monkeys. They didn't infect the monkeys. Over time, the monkeys caught the virus from the pigs and died. So they said, okay, we were very careful. We made sure that there was no direct contact between the pigs and the monkeys, and so it could be spreading by aerosol. It's very hard to prove that, one of the workers one day didn't grab a pig and then grab a monkey and spread it that way. So they're in the same room for a couple of weeks, so maybe there is some possibility of spread that way. The same group, though, and this doesn't make the papers, the same group, the same year published, they infected monkeys with Ebola Zaire, put them in cages, and the cages just have bars on them, so they're fairly open. Put another group of monkeys 1 foot away from the first group of monkeys and didn't infect them with the virus. The uninfected monkeys did not get any virus from the monkeys that were 1 foot away from them over a period, I think, of a couple of weeks. And monkeys aren't terribly nice. They like to throw things at other monkeys and whatnot, and still the virus didn't spread. So this isn't to say that Ebola can't spread through the air. We just have never known it to in human populations. So that is low on our list of concerns. Again, I'm a scientist. I'm never going to say never. But right now, we don't have any evidence that it's mutating to be spread by air. And there's some evidence to support that. So what are the treatment options for Ebola? Well, right now, it's simply pretty basic. But before I go a little further, I have asked some people the difference between a drug or a therapeutic and a vaccine. So typically with the vaccine, we use it to prevent disease, usually. So you vaccinate people you think might get the disease, and you generate an immune response to block infection. whereas a therapeutic is generally targeted for individuals that are actively infected, and it targets the virus, not your immune system in general. So the current treatment is basically very simple supportive care. Throughout infection, these individuals, of course, they lose a lot of fluids with diarrhea and vomiting. You try to give them fluids orally. It usually doesn't stay in. It usually gets vomited out. So you can give IV fluids, and that can help. Electrolytes to make sure that you have the proper electrolytes in your system, which can regulate blood pressure and other parts of your physiology. And sometimes you can get a bacterial infection after you've had Ebola. So you have Ebola, and then you get a bacterial infection. And so some of these bacterial infections can be treated with antibiotics. So we don't have conclusive data that this helps individuals survive, but it probably is somewhat effective in controlling disease. And I will talk briefly about some experimental therapeutics that are being tried in the field, and especially the ZMapp that people have probably heard of, which is antibody therapy, and some experimental antivirals, which we'll get to in a bit. Any questions so far? Okay. So the current Ebola outbreak, this is West Africa. What this map shows, this is up to date as of a couple of days ago, October 20th. This map shows anything in red is the proportion of cases that are within the last 21 days. Anything in gray is the proportion of cases that began prior to 21 days ago. So if you see something that's totally gray, it means there haven't been any new infection in these areas in the last 21 days. If you see something totally red, it just means within the last 21 days, all the infections have just begun. So you can see there's some places where the virus is fizzling out, there's some where it's spreading. And you can see some areas, such as here in Freetown, where you it's been going on for a bit, but most of the cases are actually new. Of course, the virus has been exported to Spain from sending individuals that were working in West Africa. They got sick, sent back to Spain. Two priests were sent back to Spain, both succumbed to the disease, and one of the nurses got infected, but she appears to be recovering. And then, of course, the Dallas patient who flew from Liberia and ended up transmitting the disease to two nurses. And as of last night, a doctor treating individuals in West Africa in New York City has come down with the Ebola virus. So here's another, it's a similar map, but it's a little broader, confirmed Ebola cases around the world as of 2 days ago. I mentioned the Mali case, that just happened. It's not terribly surprising since it's on the border. We're going to talk about Nigeria in a bit, and they've done a really good job of containing Ebola there. And then the Democratic Republic of Congo has an Ebola Zaire outbreak, but it's completely different from this current outbreak. So it wasn't spread to the Congo, or to the DRC, but they tend to have sporadic Ebola virus infections. So there's one going on there right now, and there's about 38 cases. And they know that it's not the exact same virus, it's the same type of virus, but it's a little bit different. They can tell that by sequencing the virus genome, which with technology today they can do in a matter of days. So I like to say that, I think we need to be honest about the relevance of the outbreak as far as it pertains to widespread in different places. In West Africa, to be honest, is really bad. This was a prediction from September 22nd saying about how many cases there would be by October 26th, and it estimated about 14,000. And right now we're right around just above 10,000. The people that I'm talking to are saying it's probably more than that. So the amount of cases and how quickly it's growing in West Africa it's spreading really, really, really quickly. It's not increasing in spread as far as the virus that we know of isn't becoming more dangerous. It's just a numbers game because it's in such crowded areas that it's just spreading very quickly. So there really needs to be increased global support for building field hospitals, for bringing in mobile laboratories that can basically test to see if people have Ebola, for bringing in home kits that they can send to people that if the hospitals are full, you can have protective clothing for the people in the homes that are caring for their loved ones that are sick with the virus. There's a dire need for that. So it is not being contained right now in West Africa. In the United States, it's not so much of a problem, or really anywhere else in the world other than Liberia, Sierra Leone, and Guinea at the moment. But we do need to put things in perspective. This is a leading causes of death in all of Africa. So it's not just West Africa, but all of Africa. So you have to keep that in mind. in thousands over the time of the Ebola virus outbreak. And you can see Ebola is not nearly as, it's not affecting the continent nearly as much as HIV, malaria, diarrhea. That doesn't mean we don't need to be concerned, but the number of cases is dwarfed by malaria cases. What we have to be concerned about is the increased spread of the virus, which is not being contained right now. So the current Ebola outbreak in the US, there's no reason, it's fine to be concerned, there's no reason for panic. This is from September 30th to October 8th. I think right now we have 1 death of Ebola in the US, and you can see from unintentional falls during that time period, we've had 602. So lots of cause for concern in West Africa. There's, in the US, I think we have, I think we need to take care of the individuals that are infected. We need to be really careful about tracing and isolating contacts. But our concern, I think, needs to be trying to contain the spread in West Africa. So there's some good news. I like this. This is a success story of the current outbreak a few months ago in Nigeria. So this is Lagos, Nigeria, a city of 21 million people. I have a lot of good friends from Lagos. And you can see it's a very crowded city in certain areas. So obviously if someone's going to be infected, it's easy to spread the virus very quickly. So there's a gentleman from Liberia that came down with Ebola virus. And against the advice of the hospital, he left the hospital and he boarded a commercial jet with 70 some odd people on it and flew, making 2 or 3 stops along the way, from Liberia to Nigeria. He vomited while he was on the plane, sick of Ebola. They found him after his arrival in Lagos, again, 21 million people in Lagos, hospitalized him. He was initially treated for malaria because he said, no, I've had no contact with Ebola. So again, the symptoms are very similar to malaria. So it's like, okay, it's very common, treat you for malaria. It didn't work. A few days later, they diagnosed him with Ebola virus, and he died soon afterwards. He contacted almost 900 people from the time he left Liberia to the time he was hospitalized and during the course of his hospitalization. Of those 900 people, 20 people got infected from the 900. Not a single one of the infected people was on that plane. So here's a man who's violently ill with Ebola that vomits on a commercial jet that everybody's sharing the same air, 70 people on it. No one from that airplane got sick. So the airborne spread, I'm not saying it's not possible, it just doesn't seem to be the primary cause of transmission. So what Nigeria did is they did an excellent job of isolating the patient, tracing his contacts, and they made over 18,500 face-to-face visits with those contacts. And what they do is like, okay, you were in close contact with this individual while he was sick with Ebola. They take temperature every, twice a day, once a day. They follow these individuals. If they spike a fever, which is the first general sign of Ebola virus infection, then they isolate them and test them for Ebola. If they're negative, you can treat them, but you don't have to worry about Ebola infection. And because of that, Nigeria is now Ebola-free. So the last case has passed the 21-day deadline, and from a man that was violently ill that flew into a city of 21 million people and contacted almost 900 individuals, this is remarkable. But it shows that the virus is not going to spread like SARS or even influenza will. So here is, very quickly, a chart of that outbreak. So here's in July 17th, the individual was confirmed with Ebola, and you can see about 10, 12 days later, incubation period of the virus, right? Between 2 and 21 days. So in this case, it tended to be about 10, 12 days. There's a new batch of people that got infected, and then there's another couple of individuals that got infected, and it finally petered out. So here's another way of looking at it. Here's the index patient, and one here is all the people that got sick directly from the patient. And then the secondary transmission from people that were infected from the index patient, and then 3 people received it from the secondary transmission. This individual also traveled by airplane while sick from Lagos to Port Harcourt in Nigeria. These 3 individuals became sick from this individual in Port Harcourt, but nobody that was on the plane with this individual also got sick. So, and the last infection was diagnosed September 1st. So it's pretty remarkable, but this virus can be contained by simple isolation and contact tracing. And what I'd like to point out is that just recently, Nigeria instituted a polio eradication drive, basically, where the government set up to try to eliminate polio there. And so this infrastructure had recently been in place to be able to track individuals, to be able to administer care to them, and they used this infrastructure for the Ebola outbreak, and they were remarkably effective. So, update from the what's going on in Dallas. From this man, Thomas Duncan, is Liberian. He was not showing any symptoms, and he flew from Liberia to Dallas. A few days after he got into Dallas, he had a fever, and he went to the hospital because he was very sick. And apparently, from what you hear reported, which isn't always accurate, apparently he said, "Yes, I've been to Liberia," to one of the healthcare workers. That didn't get transmitted to the doctor, so they sent him home with antibiotics. A couple of days later, he's sick again. He comes in, then they find out that he does have Ebola. Unfortunately, he passed away. And he contacted 48, his original contacts were 48 individuals. And as of a few days ago, 43 of them had been cleared. They never developed Ebola symptoms. I'd like to point out that his family and the people that were living with him in his, I believe in his apartment are within these 43, so none of them received it from him. So they were living with an Ebola patient, and again, according to the reports you hear, the bed was covered in vomit, and it wasn't pleasant. None of them developed Ebola. There's 5 more pending clearance. These are the 2 nurses that two of, I believe, 70 some odd individuals that were treating this man. They did come down with Ebola. The good news is this lady has been upgraded from fair condition to good condition. This lady was declared Ebola negative as of 2 days ago. They're still monitoring her, but she doesn't appear to have active infection. I'd like to point out this is one of the American doctors that was flown to Emory Hospital in Atlanta, and where he recovered, and he's donated some of his plasma, which we'll talk about in a bit, to this lady, and that she survived. So whether or not it was effective, we don't know, but there are some studies ongoing with that. There's 120 other contacts with this individual that are being monitored. If none of them come down with infection by November 7th, this outbreak will be declared over. So one of the questions I get, and this delves a bit into politics, which I don't really like to get into, but on the scientific side, why isn't there a vaccine? Well, there's some challenges with working with Ebola virus. This is a dork here in a blue suit. It's me, for those who can't tell. At the U.S. Army Medical Research Institute of Infectious Diseases. These guys, yeah, they're about $3,500 a piece, and we go through a couple of them a year for every individual, and we're really high-tech. We call them blue suits. They're made by the same people that make the ILC Dover, that make spacesuits, but it's a heavy plastic suit. It covers your entirety of your body. In order to work with live virus, you have to use a BSL-4 lab, or Biosafety Level 4 lab. It's the highest level of security there are. There's only 5 operational right now in the United States. USAMRIID in Fort Detrick, Maryland, where I worked, the CDC in Atlanta, the National Institutes of Health has a lab at Rocky Mountain Laboratories in Montana. There's a small private lab in San Antonio, and then one at University of Texas Medical Branch in Galveston. There's a couple more that have been built that they're safety testing now, 2 more in Maryland, 1 in Boston, but to my knowledge, they're not quite yet operational. It's very expensive to work in these labs, and there's some limitations on equipment. They don't like you to bring in glass unless you have to, because you don't want to slice anything and cause infection. Anything that you bring in there has to be like equipment. So I had a piece of equipment that is about $200,000, and it was necessary for me to do my work. But if something goes wrong with the equipment, you have to get it out of the BSL-4 suite, and that causes, you can do that with paraformaldehyde gas, which is harsh on some equipment. So there are some technical limitations. There's a lot of security checks. You have to go through training. So right now, depending on where you are, it takes about a year from the time you start working until you can go into BSL-4 by yourself. Depending on who you talk to, it can cost hundreds of thousands of dollars for the FBI to do a background check on you, to make sure there's nothing untoward, for all the training that you have to go through during that year. So it's a very rigorous process. If you're working with the virus, of course, you have to be super careful. Animal handling, I mean, we do animal testing. We do a lot of cell work as well. Anything in a test tube that we can do to narrow things down, but then you have to test in animals to see how effective it's going to be. So that can take time, and it's technically demanding because you're in this bulky blue suit and you're trying to inject animals, and there's needles and there's things that bite, and it can be tricky, and there's a ton of paperwork. Oh, the bureaucracy of it all. So real briefly, the way it's set up as you can, and it varies a bit from lab to lab, but in general, once you get all your training and your security passes, you enter into the room. And all of the BSL-4 suites are under negative air pressure. What that means, if there's a breach in the wall or a window, air will rush in, and so nothing in the air will get out and contaminate the rest of the room. So there's a series of doors you go through. First, you pass it, you put in a PIN number that opens the door, and so you go into a change room, and you strip down your birthday suit and put on scrubs, and then you put your you do a fingerprint scanner to make sure it's you, and do another PIN number, and you go into the next room where you put on this blue suit. There's other suits too. There's some Delta suits that were developed by the Russians that a lot of people like. And so basically you put medical gloves on your hands, and then you tape them to your sleeve of your scrubs. You pull up socks over your scrubs. You look really cool while you're doing all of this. You can't have any piercings. There's some uncomfortable stories about that. And then you get in this blue suit, and the blue suit is like heavy plastic, and you zip up, and you're completely covered by the blue suit. What you see here, it's kind of difficult to tell, but you have to put gloves on your suit, and you tape them with basically electrical tape to make a seal. And these gloves are kind of like what you'd use to wash dishes with, you know, the old dishwashing gloves. So it's kind of bulky. So you have your normal gloves, you have heavy gloves over it, and then you zip up, and you can hit another PIN number to finally go into the BSL-4 suites. Well, so you're in this suit, you put boots on over your suit, and what you have, of course, you have to breathe somehow. So this little yellow tube here is an air hose, and it hooks up to a filter that filters the air as it goes in. And the air comes into your suit, and it goes out 5 different places. There's one blast of air that continually comes from your head so you can breathe. There's air that comes inside your suit right at your hands, and also right at your feet. Now, of course, the air has to get out somewhere, so you have vents that are on each of your knees, and then 2 vents on the back. So you kind of poof up like a pillow a bit. And so you're walking around with this. And so, you know, the work is very difficult. It's very rewarding because you feel like you're doing something that might be able to help people eventually. But it can be very difficult. And then when you're done working in the BSL-4 suite, then you enter into a shower that sends a disinfectant chemical all over your suit, and you wash and you scrub down, and then water comes down from the shower and gets out of your suit. Then you waddle out, you remove your suit, you hang it up, you take off your scrubs, you go into another set of doors, you take a normal shower out, and then you're finally out. So unfortunately, people tend to share. So you know, some of these places, it's, you know, people coming in, people going out, and sometimes people forget to turn on the lights, and that someone's in here, someone's not. So there are encounters with naked scientists, which I can tell you, not a pretty sight. So one of the vaccine work, the virus was first discovered in '76. There's not a lot of labs that do the work. There's a lot of time and expense involved, so it can be very difficult to do the work. However, there are Ebola virus vaccines that are in development. I want to make sure I have plenty of time for questions, so I'll get moving here. I won't go into the details of all the different ones, but there's three that I'd like to point out. There's one, this is the Canadian developed vaccine that was tested at USAMRIID. It's called VSV. There are human trials underway right now, and this is the VSV. It's basically a non-pathogenic virus called vesicular stomatitis virus that's been engineered to carry the Ebola GP gene, the Ebola GP protein, and you put it in the body, and it makes an immune response against the gene, or against the Ebola protein, and it can protect against infection in non-human primates. The VRP vaccine protects as well. The other one that's undergoing human trials from the NIH is adenovirus vaccine. It's basically they've engineered a virus that causes colds, and they used a type that causes colds in chimpanzees, not in humans. And they made it so that it can't replicate, and they put an Ebola protein in it, and so that it looks like this, and that is also under trial, human trials. The virus-like particle vaccine is one that was developed in the lab that I worked in, and so I didn't develop it, but I did some studies with it, and it's a safe vaccine that works in non-human primates as well. Okay, so what work am I doing on Ebola? I will do just very briefly. This virus-like particle vaccine is an empty virus shell, so it looks like the virus, but it's not infectious. And so it's been shown to be protective in monkeys. So my job was, okay, if you infect animals with Ebola and you give it after infection, can it be protective? Very long story short, if mice do not receive, if they get infected with Ebola, they die. If they receive this vaccine 24 hours after infection, they live. So when I was at USAMRIID, I looked at what are the immune mechanisms by which the virus induces protection. I won't go into nitty-gritty detail. Basically, I found a subset of immune immune cells and immune products that were required for protection, and a list of those that were not required. And again, this is not to get too technical, but in typical Ebola infection, you get a lot of inflammation, you get a lot of virus replication, and that leads to impaired immune responses. And what we found is that if you treat animals with virus-like particle vaccine, you get very good immune induction, which decreases Ebola replication and leads to very effective immune responses. So I'm now working on a vaccine development. So part of the issues that we have is, in the past it's been difficult to test this vaccine in humans because before this outbreak, so few people get infected that it's very difficult to say, hey, let's do human trials on 100 healthy individuals when there's very few people getting ill. So there hasn't been a lot of testing in humans. It's very expensive. And one of the questions is, do we want to spend money on Ebola or something like HIV or malaria, which infects millions of people a year? So there's those considerations. There's safety concerns for many of the vaccines. The typical way to make vaccines hasn't been effective in Ebola, so people have had to use kind of odd platforms for making Ebola vaccines that haven't been used in humans before. So those are some of the issues as well. And stability, does the vaccine work once you take it out of the refrigerator and it's in 100-degree weather for a couple of days while you're transporting it to a remote location? And also remember I said there's 5 types of Ebola, 4 of which are dangerous in humans. Most of the vaccines are engineered against single Ebola viruses. So my job is to improve Ebola virus vaccine efficacy. So we don't have a BSL-4 lab here at UNM, but there's a lot of tricks we can use. We can use non-infectious pieces of the virus. We can vaccinate animals to measure their immune responses without having to use live virus. I do have collaborators back at USAMRIID, and we can vaccinate animals and find good immune responses, send them to USAMRIID, and they can challenge them with live virus. And we can do a lot of test tube studies to measure how potent the Ebola virus vaccines are. So I'd like to get to some of the therapeutics, and we can talk about my work a bit later if anyone's interested. But one of the projects in the lab is how sugar patterns on the glycoprotein, or the GP of Ebola, which is the surface protein of Ebola, affect vaccination. So basically, we're modifying the Ebola virus glycoprotein, which is used in every single vaccine, to try to get more potent immune responses. So we can change it based on what's on the surface of the protein versus the sequence of the protein, the type of the protein, to try to regulate better immune responses. Pretty pictures showing some of the technology that we use. We can identify multiple cell types, see how they respond to the vaccine, but we won't go into that. The second project we have in the lab, remember I said, is most vaccines are toward only a single type of Ebola virus. So we're working with individuals at USAMRIID and at the Los Alamos National Labs to generate a glycoprotein that is basically a fusion of the many different types of Ebola virus to try to generate a vaccine, one vaccine that will cause immune responses against all of the virus, all different Ebola viruses. And that's a collaboration that started at USAMRIID and is also with Los Alamos and here at UNM. So with the last few minutes, I want to talk about the therapeutics. So if you remember a month or 2 ago, there was this much hubbub about something called ZMapp treatment that was used to treat a few patients. So basically what it is an antibody treatment. What antibodies are is they're proteins that are made by the immune system when you get infected. And the proteins are very specific for what they're made against. So Ebola antibodies that are generated after Ebola infection are proteins that bind to Ebola and Ebola only, and they neutralize the ability of the virus to work. So one way of doing that is, so here, this is probably a bacteria with what appears to be legs, but so for example, say this is a virus, work with me, it's a virus. This is a virus that can bind to and infect human cells, but after you get infected, your body generates immune responses, and if you overcome the infection, the immune responses stick around. So this is an antibody. It's kind of shaped like a Y. It sticks to the virus, and it blocks it from being able to enter cells. So what individuals did is for the ZMapp, they made antibodies like this. It was a collection of 3 different types of antibodies, mixed them together, that binds to Ebola and stops it from being able to get into the cells. So that's what these individuals received. And I wanted to tell you briefly the story of how this happened, because you hear it in the news, but you don't hear the long, tortured history. So there was earlier outbreaks. There's some evidence that serum transfer in humans offered protection to those that were infected with the virus. So these antibodies that are generated from people that survive infection live different places throughout the body, but at high concentrations in the blood. So you can isolate serum or plasma from the blood, and this contains the antibodies against Ebola. So in the early days of Ebola infection, they'd find someone that survived Ebola, they'd take their blood, they'd basically remove the blood cells from it and take the antibody-rich serum or plasma, give it to someone that was infected with Ebola to cause protection. There's some evidence that that worked, but there weren't a lot of data to support it either way. So in the mid-90s, Russian scientists got vaccinated horses and got a ton of immune sera that had antibodies against Ebola. They put them in baboons, infected these baboons with Ebola, and they protected the baboons. So they sent the same batch of antibody-rich sera to Usamrid, who infected macaques, which is a rhesus monkey or a cynomolgus monkey, that were infected with Ebola. They did not see protection. So they said, okay, this is garbage, it doesn't work. There's also, not to get technical, but instead of serum containing antibody, you can use just individual antibodies. There's one antibody that was derived from a person that was infected and survived Ebola, so they made a big batch of antibody. It protected rodents from Ebola. It didn't protect monkeys from Ebola. So there's a couple of investigators over a decade that would harp on and on and on about the uselessness of this type of treatment and how it wouldn't work. People were mocked for trying it. They're like, oh, this will never work, this will never work. And then fortunately, one of my friends, he's one of the really good scientists and really good people in this field, he showed that if you vaccinate monkeys with Ebola and you take the serum containing the antibodies, and you purify the antibodies, put it into the monkeys and then infect them with Ebola, completely protective, even if given 48 hours after infection. So here's his study. These are monkeys that died because they didn't receive the antibodies from the serum, and these are monkeys that lived. So you can see that the protection was completely efficacious. So ZMapp is basically a cocktail of 3 different antibody preparations mixed together, and it's been shown to protect monkeys up to 5 days after Ebola virus infection. Now, how effective this is in humans, we don't know. A total of 7 humans received ZMapp. 5 of them survived, but all 7 of them received very intensive supportive care that's not present in lots of places in West Africa. So, and then they ran out of supply because it was an experimental drug, and they're trying to generate more. So that's the ZMapp. Real briefly, there's other experimental treatments that work against Ebola virus in monkey studies. They haven't been tried in humans, but you might hear of a Tecumseh having a drug. And basically what it does, it's a piece of genetic material that degrades the virus genes. So that's one that may be tested. There's another way you can do that with something called PMOs that blocks virus gene expression. That might be tested in humans. There's a couple of drugs on the market that that have anti-clotting activity. They haven't been very effective in monkey studies. So concerns about the existing therapeutics, of course, they're experimental. There's safety concerns. You never know how it's going to work in someone that's sick. Most treatments only target one of the 6 different dangerous Ebola viruses and Marburg viruses, and some treatments can be bypassed by viral mutations. So future directions for Ebola virus research, I think this antibody-based research, the ZMapp, what people are starting to do now is they're taking serum, like I said, from people that survived Ebola and transferring the antibodies to people that are sick. I think that research might be very effective, and there's going to be a lot more studies on that. And also development of vaccines that work against multiple types of Ebola virus, and then developing drugs that block the ability of the virus to replicate. And then one of the things is you actually can generate an immune response even in an infection where someone dies. Sometimes they still have a decent immune response, it's just too late. So one thing you can try to do is control the dangerous part of your immune response, the overinflammation. Try to control that so it doesn't damage your body so that your immune system can wipe out the virus. So I expect some research for that as well. I'd like to thank a wide range of people. DJ Perkins is my supervisor at UNM. He's the director of Center for Global Health in the Department of Internal Medicine. In the last year, he's given me a lot of leeway to write grants and start this work, so I'd like to thank him for his help. It's been invaluable. Ravi Dravasula is also at UNM Center for Global Health. He was chief at the VA, and now he's going to be director of the infectious diseases in the Department of Internal Medicine. He's the first person I contacted about a position, and Dr. Mosley is the chair of internal medicine. I'd like to thank him for his support. There's a couple of administrative staff, Marissa Sanchez, Danielle Rapella, and Jenny LeBlanc. They don't get the credit they deserve, but they've really helped me in the grant writing process. And all of this stuff, it has to be funded from somewhere, so they've been invaluable here at UNM. The work I talked about wearing the blue suit at USAMRIID was all done, most of it was done in Sina Bovari's lab. He's quite an individual. When I was there, he had 40 some odd people in his lab. He had 6 kids at home, so he was running all over the place. He was very good to work for, and he gave me a lot of leeway in the work. And I wanted to thank Kelly Stuthman, Sean Van Tongeren, and Jay Wells. These are the animal technicians that did a lot of the BSL-4 work. I would do some animal work, but they did a lot as well. They can do anything in BSL-4 that you want them to any type of animal. They're very humane. They're very hardworking. They put their lives at risk every single day because all it takes is one needle stick, boom, you might be dead. And these individuals did it without complaining, and they are very, just great people. Keiko Ozato, with the virus-like particle experiments I talked about briefly at the NIH, is one of the smartest scientists I know, over 350 publications, and one of the most humble people I know. So I'd like to thank her for her help. Richard Hotchkiss, I didn't talk about these studies, but he's a sepsis MD at Washington University, a great individual we worked with. Mike Bray and John Dye. And these are 2 individuals, Precaution Zach, that are in the Center for Global Health. And poor Zach, I sit right next to him, and he puts up with me day in and day out. And then I'd like to, we can do lab work and talk about that, but there's individuals that are actually actively working with patients. This is a gentleman who's one of the premier Ebola doctors in West Africa that contracted the disease and passed away. So there's a lot of people on the field that are doing much more important work than I am. I'd like to thank them for their support, and our thoughts and our prayers are with them. So at this point, I'd like to take any questions. I would like to say, if you want to find out more from Ebola, cdc.gov has the good information. The World Health Organization does as well. And then this is my email address. Please feel free to email me if you have relatives or friends that are interested about Ebola and they want to ask questions. Have them email me. I mean, this is part of my job. A lot of our research is funded through public money. And so I think it's important for us to contact individuals, let them know what we're doing, whether what they hear on the news is correct or not, and then you make your own decisions from the information. So thank you for your help, and I'd be happy to take questions. Questions? Yes?
How fragile is the virus in that you showed bleaching boots and gloves?
Yes.
Does that work 100% of the time if they actually cover it?
Yes.
And cremation, is that?
Great question. Yes, both of those. So the virus it can stay on, and the research on this, there hasn't been a lot done on it. It can stay on surfaces probably for a couple of days. It's not the most hardy of viruses. It's an envelope virus. Bleach is very effective in destroying the virus. Cremation is very effective in destroying the virus. But those are really good questions. It's not like, for example, anthrax can make spores and they can live in the ground for dozens of years. Ebola is not that stable. But bleach is effective. Hand washing is effective. Yeah. But the typical people will, especially in resource poor areas, people will use bleach rather than some of the fancier 10% bleach, and it's effective.
So soap and water probably?
Soap and water, yeah. So what we'd always do is, like when we shower out, it's soap and water. Yeah. And you worry about bleach being pretty toxic on your skin. Alcohol-based sanitizers might be effective as well. But yeah, it's not the most stable of viruses. Yes, sir.
There was a gentleman that used to work he had a dual appointment at UNM and also at the VA, Toby Merlin.
Okay.
Works now at CDC, but he was back here now because he wrote an editorial, and I think he also gave a presentation about his thoughts about containing the virus. He seems to think that really the majority of the effort from the U.S. should be directed at Western Africa,
Right.
containing the virus over there.
Yes.
Setting up
Yes.
hospitals, doing everything we can to prevent it from coming over here, rather than spending a lot of money up front trying to deal with it once it does arrive over here.
Right.
And I couldn't agree with it more. It seems like there's really some reluctance to close off and shut down commercial air flights from those 3 countries, with the thought that maybe that would also include humanitarian efforts, military efforts to get folks up there to deal with the outbreak. And I just don't understand that. It seems like we have so many ways to get folks over there and do the humanitarian efforts that so many folks want to participate in. And our military has all their aircraft to get over there. Why not prevent the commercial flights from coming over?
So I'll preface this by saying I'm a scientist. I'm not a policy guy. I have my opinions on that, but I'm not an expert in it. I agree with that it's important to contain the spread in West Africa. And that is, so some of the soldiers that have been sent over there are CBs or combat engineers, so they're really good at making field hospitals, making infrastructure, and so I think that's really important to do that. The way I see things, and again, this isn't my expertise and I'm not making any opinions here, but I think there's reasonable compromises to be made by differentiating between people that are traveling for recreation versus people that are traveling for medical reasons. That there's going to be a lot of national discussion. The gentleman that came back from Guinea, I believe, and then went to New York City and came infected with the virus is a guy with Doctors Without Borders. So he was over there helping people, and he came over and started showing Ebola symptoms. You can argue whether or not he should have, had, been in a holding area for a couple of weeks. I don't know. Those are going to be answered. But I think people should be able to come up with a reasonable compromise. Not everything should be an either or. I think that there's plenty of room for compromise in that. But I agree that we need more resources for containing the spread in West Africa, because it's it's logistics and it's infrastructure, and it doesn't take really high-tech stuff to contain it. And I think it needs to be a global response, and I think we got to pick it up. Great. All right. Thank you all for your attention. Appreciate it.